So before we get started, if you look at the comments down below in the YouTube video, you're going to find a link to a Google Doc. This is a workbook that I recommend you keep open and working through as you look through this video. It also has a question that we would normally take up, but it's something you can answer within the Google Doc and something we can take up in a future lecture. Now, how I want to start off this mini lecture is to look at where we're going to look at action potentials and we're going to look at cell signaling. But first, we're going to just briefly look at the anatomy of a neuron. So a neuron, very much like other cells, has a body, a soma, as well as a nucleus where we've got, DNA, we produce our RNA. But unlike other cells, we've got these unique components. One being dendrites, right, which are these tendrils that reach out towards neighboring neurons, which they're going to receive a signal in terms of a chemical messenger known as neurotransmitters. And that's going to start the production of an internal signal within the neuron called an action potential, which we're going to talk about in a couple slides. And action potentials are these brief changes in or variations within membrane potential that can propagate its way down an axon. And it's going to propagate its way towards our axon hillock. And that's where, if the action potential survives and is strong enough, it's going to elicit neurotransmitters to be released from the axon terminal and then get released and either interact with a neighboring neuron across what we call a synapse, or maybe it's a neuromuscular junction or for like a skeletal muscle. Now, that action potential is going to get generated within the axon, within something called the axon hillock. And it's going to move its way down here along this axon that's surrounded by Schwann cells. Now, Schwann cells are glial cells which provide nutrients for our neuron, but they also produce a membrane which is called a myelin sheath. Now, a myelin sheath, what it does is it insulates our axon, because our action potential, when it gets generated here, is going to basically elicit electrical current that goes down and gets basically rejuvenated at these points called nodes of Ranvier. So it's sort of like it's bouncing its way along, because those are the points that allow for the exchange of ions, which is very important for our action potentials, as we're going to see in a moment. So how cells or neural cells talk to each other is through the process of cell signaling. So here we've got basically two adjacent neural cells, and they are separated by something called a synaptic cleft. And so here we've got the axon terminal of the one neural cell, and then we've got the dendrite of the other neural cell. And so in this, we've got a small little window called the synaptic cleft, which is very, very tiny. It's 20 to 30 nanometers in size, which, for comparison's sake, the width of a piece of paper is 100,000 nanometers, or a piece of hair is like 75,000 nanometers. And so it is a very, very tiny little sliver. And it is across this synaptic cleft that we are going to release a bunch of neurotransmitters to create a signal in the neighboring dendrite. And so our neurotransmitters are getting released from the axon terminal, and they're going to bind to specific receptors that are unique to those neurotransmitters. So some neurotransmitters like GABA or dopamine, right, they're going to bind to very specific receptors from those neurotransmitters. Now, those neurotransmitters aren't going to stay very long within the synaptic cleft. There's always like a small concentration within there, but a lot of them are going to be reuptaken into the axon terminal of the neural cell so they can be used. And they get stored in these small little containers within the axon terminal called vesicles. Now, the neurotransmitters get picked up by neurotransmitter transporters, which are specific to those neurotransmitters. And they can be like symporters, where basically sodium and potassium molecules will interact with their protein structure and allow them to take up neurotransmitter by using ATP. Some neurotransmitters will remain within the synaptic cleft and can get broken down by enzymes, right? So acetylcholine gets broken down by acetylcholine esterase. To understand how neurotransmitters can create an action potential within a neural cell, we need to discuss something called membrane potential. So an action potential is a rapid change in membrane potential. But what is membrane potential? So here is a pretty simple diagram. Here we've got a membrane, and in the membrane we've got our intracellular and extracellular matrices or regions, and then our membranes are, our phospholipid bilayer. And the resting membrane potential is the voltage across the membrane. So remember, our cells have ions within them, and those ions have charges. There's positively charged and negatively charged ions. And if we can separate the charges out so that there are more positive ions on one side and more negative ions on another side, then there's going to be a difference in voltage or difference in potential. And this is going to cause the ions within those to want to try to move, right? Because the positive ones are going to repulse other positive ones, and they're going to be attracted by the negative ions or anions. And the phospholipid bilayer is going to prevent them from going back together and forming some type of equilibrium. And so the more disequilibrium there is, the bigger the potential, or the bigger the voltage. So when we look at these different ions, they have that electrical gradient, is what we call it, right? That difference in charges between the two different regions that really wants to reach equilibrium, but it can't because it's got this barrier in its way. Also, there's a concentration gradient, right? So there's going to be different concentrations of ions on one side versus another. And so if we have a lot of sodium ions on one side and very few sodium molecules on the other, the sodium wants to move down that concentration gradient. And again, it's being prevented by that phospholipid bilayer. So here we've got our extracellular matrix where we have a lot of sodium and chloride. And then in our intracellular, we've got a lot of potassium. And so this creates a concentration gradient, right? Potassium wants to get out of the cell, sodium wants to get into the cell, but it also creates a electrical gradient because there's a difference in the number of charges between the two different areas. Now here we've got our ion channels. They're closed. They're going to prevent the movement of ions between them. And this creates a resting membrane potential of negative 70 millivolts. The positive is on the extracellular side, and our negative is on the intracellular side. So you notice that we've got a lot of positive ions on both sides. So the difference has got to be made up somewhere. And that difference is peptides and phosphate ions that exist within the intracellular region, right? They represent the overwhelming negative charge that we see within the intracellular space. Let's talk about action potentials. Now that our neurotransmitters have bound to the dendrites, they will now cause changes within gated channels along that membrane. And these are going to let in sodium ions flood down their electrochemical and concentration gradient into the cell. These are ligand-gated channels at the start. And if there's sufficient amount of excitatory signal being sent to the axon hillock, this can generate an action potential. It has to reach basically a certain threshold. If it doesn't reach that threshold, then essentially there's going to be a bunch of false starts. And so as sodium is flooding into our intracellular region, this will cause depolarization of our cell. We'll go from negative 70 millivolts all the way up to plus 30 millivolts. Now, once we pass that negative 55 area, we will begin to open up a whole bunch of other voltage-gated channels, which starts that positive feedback loop where we get more and more sodium pouring in, which causes the intracellular area to get more and more positive, resulting in more and more voltage-gated channels being opened. If we don't reach that threshold, we get a bunch of these false starts. Now, once we get to plus 30, we begin to go through repolarization. This is where our sodium channels are being locked up by inactivation gates because they've been triggered by the now very much positive intracellular area, and our potassium begins to flood out of the system because potassium voltage gated channels open up, pouring potassium out of the system into the extracellular space. This causes the voltage or membrane potential to drop from plus 30 millivolts down to, well, it's going to go past negative 70 actually, and it's going to go to negative 75 because our potassium channels, they don't close as readily as our sodium channels, and so they leak out into the system, which causes us to go past that negative 70 point into repolarization or hyperpolarization. Now, hyperpolarization, this is a region where we have a certain period of absolute refractory period where you just cannot generate a new action potential. And then afterwards, there's a relative refractory period where if there were sufficient enough excitation to overcome the outflow of potassium channel of ions, then you might get another action potential happening. Now, once we've gone through this little lecture, what I want you to do is hopefully you've been going through that workbook, through the Google Docs, and there's some questions at the end which you can use to test your knowledge, and then we will then take that up at a future date. Thank you.